I think what I'm going to do is I'm going to talk a little bit about the basic structure of carbohydrates, okay? And in essence, this is really a very fundamental lecture. Some of you who have been exposed to carbohydrates probably already know it, but it will be a good refresher. And for those who are primarily engaged in biology, you might have forgotten a few things. So this is a very fundamental lecture that every carbohydrate chemist needs to know. At a very fundamental level, carbohydrates are very unique structures, completely different from what you see across the other biomolecules. Overall, carbohydrates are really, really important. And here I list few examples, not many, but in essence, the major reason why carbohydrates have been studied up until now, for most part, is because people thought that carbohydrates are basically energy sources, right? Carbohydrates supply a lot of energy. So they are primarily energy supply, especially, for example, so how does this work?
The front one, or the pointer.
Do you see a red dot? I think the battery is dead. That's okay. That's fine.
Is it okay moving forward as well?
That's fine. Don't worry.
Okay.
I think the battery is dead. So, so carbohydrates.
Is working, right?
I can use a cursor.
Okay.
Just relax. That's okay. I'll use the cursor. Okay. So hopefully you can see the cursor. Yeah. Okay. Oh, all right. This is the exact one that I use at my place, you know. Good, very good. Okay, so carbohydrates have been primarily explored as energy sources. All right,
It's working now. Okay.
I think it was in sleep mode.
Yeah.
I'll keep it,
yeah, sure.
just in case.
Of course.
Okay. Okay, good. All right. So they are source of energy for animals too, and in general, carbohydrates have been studied primarily from the energy perspective. Well, the second major reason why carbohydrates have been studied is because carbohydrates are primarily skeletal material. They actually supply, are involved in forming the base scaffold of, for example, plants. You might have heard a lot about these days in terms of energy and lignocellulose, for example, which is really, really important, which has picked up big time use, and how do you break it so that we can get a lot of carbohydrates, and from that bioethanol and things of that sort. Well, there's a major difference between, for example, starch and cellulose, and which is the reason why skeletal carbohydrates cannot typically be used as energy sources. And it's all dependent on structure. Structure governs whether a particular polymer is an energy source as far as humans are concerned, or is a scaffold on which an entire plant can be built. Carbohydrates are also important because they are important in terms of connective tissue in animals, acidic polysaccharides. Many of you know quite a bit about the glycosaminosglycans that play a really important role. In addition, this is something that you have already heard about. RNA and DNA have a 5-membered carbohydrate structure. And that's really a central thing. All cell surfaces have heparan sulfate. Obviously, many of you know, and this grant is all about heparan sulfate and exploiting heparan sulfate for lots of other things, for biology as well as for pharmaceutical purposes. Carbohydrates define our blood types. The 4 major categories arising primarily from carbohydrates. And these are just to prime you into the direction of these structures. This is how typically carbohydrates are written, essentially. For example, there's a letter D, which you're going to talk about. This one just skips all the time. I'll just use this. So there's a letter D, and then you have something like gal, which is the name of the carbohydrate. We have, for example, something like N, which indicates that there is a nitrogen. We have a letter P, which indicates a particular form of the carbohydrate. Then we have some things such as 2AC, maybe not 2AC, things of that sort that indicate what type of substituents may actually be present on the carbohydrate. And then a linkage as to how the two carbohydrate moieties are linked, what positions are coupled. Likewise, you might have a Greek symbol such as alpha or beta that indicates once again the type of linkage. So this is how typically carbohydrates are written. You might have a branch, and this branch you would have a linkage, for example. So in essence, all of these actually tell you what the structure is, and knowing what each of these are is important. Small differences, writing an beta here instead of an alpha, makes a lot of difference. It changes the recognition significantly, not just significantly, it might completely obliterate the recognition. And therefore, and these changes are really, really small, and yet nature has engineered specific mechanisms to be able to recognize specific structures. That's the beauty about it. That's the interesting part about it, as to how can you have small changes in orientation of specific groups completely changing the recognition. And by the way, decades of effort has gone in understanding as to how these changes are brought about. Okay. All right, let's go forward, just because we don't have a lot of time. But you can ask me any questions at any point. Okay, I would like it to be as interactive as possible. Obviously, this is fairly simple. So another major role is, for example, vitamin C. Vitamin C, ascorbic acid, is really important. Without it, we would have a major disease. And not only that, vitamin C in solution can easily be oxidized, so we have to be careful. This is something that the program project deals with quite a bit. Heparan sulfate, specifically heparin, has a specific structure. For example, this 5 residue sequence is really involved in recognizing a protein that we all have. We all have this protein antithrombin. Without it, we cannot really survive. In fact, without antithrombin, the fetus simply does not develop. So heparin is involved in recognizing antithrombin. But we don't really know why nature has come up with this particular sequence, because it appears that this sequence, for most part, is present outside the blood compartment. And yet, we have that sequence. So, how was that brought about? Not much information is available. Yet, the recognition is really, really very important. And we realistically, we expect that these kinds of sequences would be present in many other glycosaminoglycans, which is, a major research area today. All right, so starting from the very basic, carbohydrate. The word carbohydrate essentially has been used as the hydrate of carbon. So back in the olden days, they didn't really have the structure. And at that point, the molecular formula was something that people used to deduce. And the molecular formula subscribed to the idea that you would have C, N, H2O, M. And that is the reason this would be the hydrate of the carbon. For most part, that remained fairly true for years, actually for decades. And that is why all of these carbohydrates were bunched together all of these structures were bunched together as carbohydrates. Later on, it was discovered that you might not only have carbon, hydrogen, and oxygen, but you might also have a nitrogen, substituting one of the hydrogens. And that nitrogen typically is present at position 2, for example. It does disturb this molecular representation, but yet overall it is fairly similar to these kinds of structures, and therefore this particular molecular formula was also classified as a carbohydrate. We now know it as glucosamine. Likewise, you might actually have N-acetyl-galactosamine, where galactose is the base carbohydrate. Then we have an amine, and that amine has an acetyl group as a substituent. Carbohydrates typically have suffixes and prefixes. So most of the time carbohydrates end with an O's. So glucose, mannose, pentose, things of that sort. Prefixes typically represent the size. For example, tri indicating 3 and hex indicating 6. So in essence, this is how carbohydrates are typically written. Carbohydrates have many functional groups. So as you can probably imagine, they not only have an OH functional group, but carbohydrates also have an aldehyde. So when they typically have an aldehyde, they are called aldoses. Or when they have a keto, they are called ketoses. Overall, the smallest scaffold looks somewhat like this. So the smallest carbohydrate scaffold in the aldehyde category, in the aldose category, is glyceraldehyde. And the smallest carbohydrate in the ketose category is dihydroxyacetone. So glyceraldehyde and dihydroxyacetone are the smallest carbohydrates. Notice glyceraldehyde has that particular carbon which is a chiral carbon, right? Which is a chiral carbon. So you all know what is a chiral carbon. Yeah? You all know that's a chiral carbon. So when you have a chiral carbon, there are 4 different substituents attached to that carbon, and therefore that can exist in 2 stereoisomeric forms. So these are the 2 stereoisomeric forms. You can have an R form and an S form, right? You can have an R form and an S form. And the R and the S is what type of nomenclature? I know you're not going to ask me questions, so I'm going to ask you questions. What type of nomenclature is this? Where are the letters R and S derived from?
German.
They are enantiomeric, but yeah.
It's German, I think, right? It's German, right?
German.
I believe so.
Well, I'll give you a clue. 3 scientists came up with that nomenclature. Uh-oh. See, that's the reason you should ask questions, right? You don't get asked these dumb questions. Come on, somebody knows it. Okay, 3 scientists came up with this nomenclature. And that's how this is the system. If you're not familiar with nomenclature, let's call it a system. So I don't know how you have been taught earlier. It's first scientist's name starts with a C. Well, I think it's important to remember names, okay? Okay, let's try with the second scientist's name starts with an I. It's called the Ingold Prelog system or nomenclature or convention, whatever that you want to use. Yeah? Okay, you might want to write it down. Kahn-Ingold-Prelog convention. They did a great service. And by the way, this is not just true for carbohydrates, right? I hope you recognize that. This is true for any stereoisomer, right? A chiral center, unless it's a mesoform. Let's not go there. So what is R? Now, okay, I would like to confirm that you know. Because it's so simple that I'm assuming that I'm going fast and I'm assuming that you all know. So what is R? Do you know how do you define R?
Moves the plane polarized light in the right side.
Very good. So that is one category, but not for the Kahn-Ingold-Prelog convention. Moving the plane polar, I'll get to that.
It's how you go around from the, whether you go clockwise, reading through CH2, OH, CHO, whichever, I don't remember that, but it's the way you...
It's something to do with clockwise. Fantastic. We are one step closer. Fantastic. It's something to do with clockwise. So R essentially means clockwise. So what would S mean? Anti-clockwise. Excellent. So how would you know? How would you know? For example, if Prem does this in this way, we would say this is clockwise. CH2OH, OH, and CHO is clockwise, and therefore this is R. But then if you, Krishna does it, he would do this, and you would say OH, CH2OH is clockwise. Wouldn't you have a problem? Yes. That your clockwise and his clockwise would be different?
Priority of the functional group.
Priority. Is this on or what? Okay, priority of the functional group. That makes a difference. So there is typically a priority written. What is that priority? The priority is, in essence, the atomic number typically has a higher atomic number has a higher priority with that atom that is directly connected to the chiral center. So in this particular case, oxygen has a higher priority than C, has a higher priority than this C. So oxygen has priority number 1. Okay? Then the question arises, what about that carbon and that carbon? So because they have the same priority, you move one step further away. So this is a double bond oxygen, right? So therefore there are 2 oxygens here, and there is two hydrogens here and an oxygen here. So therefore this has a higher priority than that. So therefore this is going to be 1, this is going to be 2, this is going to be 3. That's how the priority is defined. Yeah? The question is, how do you know that this 1, 2, 3 are organized in an R, in a clockwise manner? How do you know? Because you know what is this, right? What bond is this? That bond is coming towards you. What is this bond? Going away. What is this bond? In the plane. What is this bond? In the plane. You would always have 2 bonds in the plane. Okay? And then one coming out towards you and one. And you have to arrange it like that. So the question is that if you are looking from this side, then that particular hydrogen is going away from you. But so it's not that, it's me. But if this bond was actually swapped with that position, right? Imagine that H is here. I should have prepared a slide like that. That H is here and this carbon is here, right? Now, priority 1, priority 2, and this being here is still priority 3. It is still clockwise. But by swapping 2 bonds, what have we done? We have actually changed stereochemistry. You all know that? You don't? You should try it at home. They actually change stereochemistry. Yeah? So how do we know?
The least priority one should always be in the plane.
Very good. So that's one of the important points. The least priority group, not necessarily H. This particular molecule has an H. H is the least priority. But in general, you may not have an H and a molecule can also be chiral. So the least priority group, you have to rotate, you have to move the molecule around so that the least priority group is away from you. And then you look at the remaining three groups as if they are in one plane. And then write 1, 2, and 3. If that is clockwise, then that is an R geometry, stereochemistry. R enantiomer or enantiomer, if you want to pronounce it like that. Right? So in this particular case, that is the least priority group. It is facing away from you. So if you look from that side, this is 1, 2, and 3, and therefore this is R. Yeah? In this particular case, that is again away from you. What have we done? What have we done? We have kept this geometry of, we have kept the orientation of that CHO the same as that. We have kept the orientation of this the same as that. And we have simply swapped these two groups. Right? We have simply swapped these two groups. So in essence, what has happened? By swapping, we have now changed priority number 1, 2, and 3. So that becomes an S, anti-clockwise. Yeah? And this logic applies for all, not necessarily just carbohydrates. Okay? Now, to address the point about plane polarized light. So, in essence, chiral molecules rotate the plane of polarization, right? You all know that. So, R and S have nothing to do with plane of polarization, have nothing to do with the rotation. They are clockwise, anti-clockwise, that's absolutely true. R is clockwise, S is anti-clockwise. But just because R is clockwise means that this molecule will rotate the plane of polarization in a clockwise direction. That correlation doesn't have. So we have polarization, optical activity, and independent component. This is simply based on structure. And the reason why this was developed was because back in the olden days, it wasn't easy to identify. Optical activity was known. People had deduced optical activity. But then what correlates with what took a long time to develop. So this was used as a tool to understand, to differentiate the two isomers. And Kahn, Ingold, and Prilog came up with this particular convention. So you might have R changing the optical rotation to left, and that would be called levorotatory. Okay? That is typically denoted by minus. And to the right, when the plane of polarization goes to the right, that is called dextrorotatory. Glucose is a dextrorotatory molecule. It's actually called dextrose, which is one of the reasons why glucose is called dextrose. Natural glucose, by the way, okay? Not the synthetic one. You can have a synthetic one which can move in the left-hand direction. All right. So this is called the sawhorse projection. This is called the Fisher projection. Okay? And the Fisher projection, you can essentially write the same molecule in this kind of a manner. The typical way in which Fisher projection is written is something like this. You always, for most part, you always have CH2OH written at the bottom, especially for aldoses. And for ketosis, you have the keto as high at the top, CHO at the top. And then you can have OH on the right-hand side, just as here, or OH on the left-hand side. Because these two positions are fixed, the two stereoisomers would only be swap of H and OH. Yeah? So this is how typically we write. And this is for the simplest unit, where there is only one chiral center. Now, the representation here equals this representation. Okay, what does that mean? That simply means that that bond and that bond are going behind, and these two bonds are going away. So there is no bond in the plane of the board or in the plane of the screen. Okay, so it's basically like that, where nothing is in here and both are. You have to convert that structure into this structure in order to analyze R and S. And you would see that both of those would be equal if you make the appropriate rotations. Remember, this is coming towards you. So you have to turn it to move it in the opposite direction. So this is how most textbooks are going to show carbohydrates. This equals this. This does not mean that the H can be considered as going behind the board. Please remember that. That is a convention. Now, when the OH is on the right hand side, that structure is called D, capital D, not small d, capital D. Okay? When the OH is on the right, that structure is called capital E. Most sugars, most, not all, most naturally available sugars are of capital D type. Okay? When the OH is on the left, that is called L. Okay? And this is a convention. So glucose, for example, we would have D-glucose. The natural L is, for example, idose or iduronic acid. These are L. So in essence, what is important here is that that carbon which is furthest away from the aldehyde, the orientation of the hydroxy, the OH on the carbon that is furthest away, chiral carbon, not CH2OH, chiral carbon, is the identifying center for D and for L. Okay? And I'll show you in just a little while. Okay, so it turns out, so this is the Fisher's projection, it turns out that D-glyceraldehyde is actually positive. And it turned out to be fortuitously correct. Okay, it could have been opposite, but he had arbitrarily assigned it, Emil Fischer, and obviously he won the Nobel Prize for elucidating the structure of so many sugars. All right, so this is what I mean by that carbon, the chiral carbon which is furthest away from the aldehyde. So this is glyceraldehyde, right? And this is the D series. Why? Because the OH is on the right hand side. So when you add another carbon, typically you add a carbon in between the aldehyde and this particular carbon. And you end up with two molecules. You can have that in the D series. You can have that, if you add this particular carbon, the OH can be on the right or the OH can be on the left. So this is D-erythrose, this is D-triose. How do you define D? You identify that carbon which is furthest away from the aldehyde, position 1, and you say this is on the right. And therefore, this is D, this is also D, right? Yeah, all clear? Because that is the identifying species. Okay, so if you had L-glyceraldehyde, and you had added that carbon and that carbon, you would have obtained L-erythrose, L-triose. Yeah? Are you all with me? So likewise, erythrose, once you add another carbon, so this is a 4-membered carbohydrate. Yeah, we are going from 3 to 4 to 5 to 6. And carbohydrates can have close to 9 carbons. Mono can typically have 9 carbons, so there can be a lot more. So when you add another carbon, that OH can be on the right or on the left. And that is called D-ribose or D-arabinose. When you add another carbon to triose, which is on the right or on the left, you would get D-xylose or D-lixose. And likewise, in the end, if you keep extending, add a carbon in between position 2 and position 1, to ribose, you would get these two. Add a carbon between these two, you would get these two. And likewise, you would get these from D-xylose, and from lixose, you would get these two. Right? So in essence, how many monosaccharides, D-monosaccharides, D-hexoses do we have? 8, right? How many L-hexoses are we going to have? You're going to have 8 L-hexoses, right? So, synthetically as well as in nature, you could have 16 different structures that differ from each other in as little as 1 chiral position or all 4 chiral positions. Okay, so you could have a difference of 1. Okay, for example, let's say glucose and let's say galactose. I'm going to highlight on this a little bit later on. So that's the reason I'm picking on these two. But you could try your own. So if you have D-glucose and D-galactose, what is the difference between the two? So that OH, okay, so this is position 1. Yeah, I didn't tell you that. So usually that is position 1. This is position 2. This is position 3, 4, 5, and 6. Yeah? So obviously this is D series, so all have to be the same because for all they are basically on the right. And then what do we have? We have position number 4. So position number 4 here in galactose, OH is on the left. In glucose, OH is on the right. Right? And if you notice, that and that is the same, and that is the same. Obviously, aldehyde doesn't remain. So glucose and galactose differ at only 1 position. Okay? Only 1 position. When we have a difference of only 1, the 2 stereoisomers are called epimers. They are called epimers, right? When you have a difference, they are called epimers. Oops, what happened? Okay, they are called epimers. When you have, likewise, you would have 1 difference between glucose and mannose. Yeah? What about glucose and gulose? D-glucose, yeah, very good. D-glucose and gulose, if you notice, you would have a difference of 2. So when you have a difference of 2, they are fairly different structures. Okay, fairly different structures. Diastereomers. This form, the way in which this is written, has a specific name. I want to ask you whether you know that name. What form is this? The structure, the way that this is written, has a specific form. A specific terminology is used. This is called the... What's that? Fisher projection. Yeah. This is the Fisher representation. Very good. This is the Fisher representation. Okay. So this particular form is called the open chain form. Okay. This is called the open chain form. Normally, well, for predominantly carbohydrates such as these, especially the aldehydes, do not exist in the open chain form. Do not exist in the open chain form. They exist in the closed chain form, and we are going to see that in just a little while. Okay, what do we have next? So, likewise, the same series is obtained from ketosis. Okay, in essentially the same series, but in ketosis, you have 1 less chiral center. Yeah? Because ketose itself, you have destroyed chiral center at position number 2. You can't have a ketose at position 1. That would be an aldehyde, right? So therefore, keto is typically at position 2. And we keep keto at position 2 and start adding a carbon in between these two. So dihydroxyacetone has no D or L form. So the first chiral carbohydrate has a 4-carbon scaffold. And therefore, you would have, the only reason why I'm drawing L is because I didn't show you L on the other slide. Otherwise, you would have D as well as L. So likewise, you would have, for example, 5, you would have 2, and at 6, you would have 4 different ketoses. Because of absence of one chiral center, you would have only 4. And likewise, for the D series, you would have 4. So that will end up with much lower structural diversity. Okay, and you can see that this is the molecule that is very commonly recognized as being very important for lots of different things.
Umesh,
Yeah, go ahead.
how did the naming come about? Like, I'm just curious, like the Xy, the first 3 letters are what about fructose, sortose? How does...
Oh, I wish I knew all of those. But I'm pretty sure after glucose was discovered that I don't know. How did they come up with the name? It's a good question. Absolutely, some story is associated with it.
Not from source of
Yeah.
Maybe from the source, yeah. Maybe from the people, a lot of different things. I don't know the full story, but it's interesting. History of carbohydrates would probably cover it. So, good, very good. Okay, so let's talk about monosaccharides. So, monosaccharides. What time is it? 5 to 10? 5 to 9. Oh. Okay, 5 to 9. So we are not even half done and we have consumed an hour. All right. Okay, let's see how much we cover in 5 minutes. All right, so monosaccharides, as we have already seen, glucose is the primary monosaccharide. Nature has D-glucose. D-glucose is the one that is recognized biologically. Enzymes recognize D-glucose. L-glucose is, interestingly, L-glucose is sweet. You actually can taste L-glucose. Okay? You can actually taste L-glucose, and it's sweet. So back in the olden days, people said, well, you know, why don't we use L-glucose as a sweetener? Because it would not be metabolized. You would gain no calories. You could put as much sugar as you want and not become fat. Well, it didn't turn out that you couldn't really use it. So L-glucose is made synthetically. It's extremely expensive to make L-glucose, and therefore you can't really spice up your stuff with L-glucose. The food would be phenomenally expensive. Okay, so D-glucose is typically present in our blood, 65 to 100 milligrams. You probably all know that. Anything above 120 milligrams a deciliter is bordering diabetes. Okay, dextrose is basically glucose and saline. Galactose is another important sugar, is part of lactose, and we already know a lot about lactose primarily because these days more and more babies are born with lactose intolerance. So, and in addition, we already said this 2-amino can be present. You would have, that's called glucosamine, manosamine, and all of these are actually present in nature. We'll see all of these. So now let's just talk briefly about the cyclic structure of aldoses, and therein lies how carbohydrates are actually named. So in general, and this is true for organic chemistry, I don't know whether you know about this, so that's the reason I thought this will be useful to add. So in general, if you take an aldehyde, this is an aldehyde and this is an alcohol. So if you take an aldehyde and an alcohol, typically what happens is you get something like this. You know what is that structure? Back in your first organic chemistry course, you would have had something like that. Do you know what is this? that is a carbon, that is an R group having a carbon bearing an OH and an ROH. It is called a, it's called a hemiacetal. That actually happens. And by the way, this is unstable. Unstable in the sense it can be easily converted back into the two constituents. Okay? So typically, this is not very stable. Obviously, if you have an R double prime here, what would that be called?
Acetal.
That would be an acetal. That would be an acetal. And an acetal is much more stable in water. We're talking about primarily in water as compared to a hemiacetal. Okay? Under acidic conditions, this would go back. Acetal would not go back. And this is important primarily because when you have polymeric sugars are going to have something like that. They are going to be acetals that are going to be formed. All right, so what about this? So in this particular case, you have an aldehyde and an alcohol, two functional groups in two different molecules. Yeah? What about this? Now you have an aldehyde and an alcohol in the same molecule. So this is intramolecular. And believe it or not, what happens is that this folds over and typically you get something like that. You see that particular carbon has an OR which is somewhat like that and an OH which is exactly like this. So this is a hemiacetal, except that this is intramolecular. It's the same molecule within the same molecule, right? Sugars are basically the same thing. Monosaccharides, for example, have an aldehyde exactly like that, and this position you have exactly like that. In between you have rest of the functional groups. It turns out that this can fold over into a conformation such as that. Notice this is the aldehyde, this is the aldehyde, this is position 2, this is position 3, this is position 4, this is position 5 and 6. The 5OH comes fairly close in three-dimensional space to the aldehyde group, and therefore the lone pair on that particular oxygen can actually attack on that carbonyl carbon to give you either that structure or that structure. So if the lone pair attacks from the top of this carbon, so to give you an analogy, a very simple analogy, imagine in 3 dimension. So if you have the carbonyl carbon in this plane, that particular carbonyl carbon, it's planar, right? sp2 hybridized. So if this is planar and that OH is somewhere here, okay, you can have the OH attack from here, and therefore what happens of that oxygen is this oxygen will go down because it's going from the top. Yeah? Are you all with me? Yet you might have a conformation such that the OH is at the bottom. So yeah, the OH, the C5OH is at the bottom. So it comes from the bottom and attacks from the bottom. Then this oxygen is going to go at the top. Yeah? Are you all with me? So in essence, you might have an OH at the bottom or an OH at the top. And you would have a cyclic structure. So carbohydrates, especially of the aldehyde type, even ketosis would give you something like that. But of the aldehyde type would not prefer to remain in an open chain form, would convert into a cyclic form. And that cyclic form can be of 2 types. Notice we are generating in essence a chiral center here. Right? We are generating a chiral center, an additional chiral center. So therefore, you might have alpha-D-glucopyranose. What is this? This is a 6-membered ring. In organic chemistry, a 6-membered ring containing a nuclear oxygen, which is this one, is called a pyran. A pyran is a 6-membered ring containing a nuclear oxygen. O's, obviously, because it's a carbohydrate. Gluco, because we started with a glucose. I didn't write here, but this is basically glucose. Okay, so it's basically glucopyranose. D, because we started with OH on the right. That's D. And now we are introducing two additional things, alpha and beta. Those are the two things. What do they refer to? They in essence refer to what is the structure around C1 position. So when the OH group is furthest, so in essence, what are we doing? We are, if this is now the plane of the molecule, the six-membered ring is not planar, okay, but imagine for the time being that it is a plane. Okay, imagine that this six-membered ring is a plane. It's not planar, and we are going to see that. It's actually a chair form for most part. So imagine that this is the case. Then, to give you a simple thing, this hydroxy is at the bottom, like that. Yeah? Where is the next hydroxy? Is at the bottom. Where is this hydroxy? At the top. Where is the next hydroxy? At the bottom, like that. Where is that CH2OH? At the top. Are you all with me? Okay, imagine in 3 dimensions. So therefore, I wish this doesn't happen. By the time we are done, we would be deaf. So this hydroxy and that CH2OH are in essence on the opposite side. So they are furthest away. And that is called alpha. Okay? You can also use the terminology that H and CH2OH is on the same side, and that is alpha. Whatever that you feel is convenient to you. I like to refer it to OH primarily because there are so many, especially fructose, for example, there is no H. Okay, and that's why it is a little bit confusing. So I like to use the OH terminology. So OH and CH2OH are away from each other, that is alpha. OH and CH2OH on the same side, beta. Yeah.
So normally in equilibrium, what percentage is in the...
Very good question. I think I have a slide. So it's coming up pretty soon. Okay? So before I go there. So that because we have introduced an additional carbon, these two are related to each other. They are anomers. Okay, so we have epimers, obviously we have enantiomers, we have diastereomers, and then now we have anomers. Anomers refer to specific stereoforms at position 1. So that carbon is called anomeric carbon. Okay, that carbon is called anomeric carbon. And this, the manner in which this is represented is called Haworth projection. So we have Fisher projection, we have Sawhorse projection, we have several of the projections, but the common ones are Fisher projections and Haworth projections. And obviously we have a 3-dimensional thing, chair form, that I'm going to show you in just a little while. Some authors like to use the 3-dimensional chair form. Some, I prefer to use this because it's all linear, Haworth. And then very, very few people use the open chain form, unless it's a synthetic paper. In a synthetic paper, we tend to use the open chain form, chemical synthesis. Okay, so this carbon is called anomeric. All right, let's go forward. Okay, so that's anomeric carbon. So likewise, and I'm going to jump here primarily because I expect you to read up. Likewise, fructose, for example, with a 2-keto, is also organized. So now in this particular case, you have a 5-membered ring because the carbonyl is at position 2 and not at position 1, that we have the same 5-OH forming a bond, and this can attack from the top or from the bottom, thereby generating an OH at the bottom with relation to CH2OH or at the top. Therefore, this is called alpha, this is called beta. Yeah, are you all with me? So spend some little time, it doesn't require a whole lot. Nature obviously has a fructose that doesn't have the oxygen at position originally. That carbon is position 2. Yeah? Notice, that is position 1. This is keto is position 2. And therefore this is position 3 as governed by carbohydrate numbering. But with RNA, DNA numbering, we call it position 2. It's 2-deoxy. That's the difference. RNA, DNA people use different numbering. Carbohydrate people use slightly different numbering. So this is when you don't have an oxygen, and this is a central constituent in DNA, for example. So we have 2-deoxy. You have a CH2OH. Okay? Are you all with me? Okay, you want to take a break or can I go forward? You want to take a break? 5 minutes? No? Go forward? Okay, so then we'll finish at 10 then. Is that okay? All right. So conformation of, so as I said, you have the 6-membered ring is not really planar. Okay? It actually exists in this kind of a form. This is called a chair. For those of you who are not familiar with chair, you can imagine that that is the back, this is our seat, and this is whatever rests on the bottom. And that is the reason this is called a chair. Okay? It's a very uncomfortable chair, but it's a chair. Okay? In essence, alpha D, what is this? This is glucose. Alpha D glucose primarily exists as a chair. Either it can be alpha D or it can be beta D. So if you look carefully, that is position 1. This is position 1. That OH is at the bottom. And then this is position 2, OH at the bottom. So these two are on the same side. Okay? Then that OH is above the plane. So you have to imagine that these 6 nuclear positions form a plane. Therefore, this is above the plane. This is below the plane. This one is at the bottom here. And it is actually at the bottom. There is an H. There should be an H here, which I have not drawn. And then that CH2OH is here, and it is above the plane just as it is above the plane. So this corresponds to that particular structure. Are you all with me? Yeah? So in essence, this is the cyclic form. This is the true 3-dimensional cyclic form of glucose. And for that matter, all molecules that can show this kind of a cyclic form would exist, with differences primarily because this OH might be here or this OH might be here, and things of that sort. Substituent effects will make a difference. Likewise, the beta D, I think as soon as I become suddenly energetic, it picks up and it says shut down. So, okay, so as soon as, So 6-membered ring with the beta OH is oriented above the plane, as you can see. So this is below the plane, this is above the plane. Yeah, are you all with me? So in essence, these are the chair conformations. Okay, just to ask you, do you know what is the nomenclature for that particular bond?
Equatorial.
Very good. That's equatorial bond. What is this?
Axial.
This is the axial bond. So how many equatorial bonds are present in a 6-membered ring?
5.
In a sugar, yeah. In a sugar, there are 5. Okay. But in general, if you have a chair, you're going to have 2 bonds here also. Okay. So those would be. So you would have 6 axial and you would have 6 equatorial. Now, do you know as to what is the preference for a substituent in terms of what is the equatorial or axial preference of substituents or functional groups? Where would you prefer to be if given a choice?
Equatorial.
They would prefer to be in equatorial. Okay, they would prefer to be in equatorial. Why? Because equatorial position is known to be less sterically hindered. Okay. So why is that? That's because the axial position is more sterical. That doesn't mean that equatorial, there is no steric issues, but the axial position has more steric issues. Okay, so for those of you who are not sure, let's see whether we have something in that direction. Can I? Okay, by the way, I can write here. Yeah? So just to tell you, and I think it's important to really know how to draw this, okay? So typically, most of you can draw very easily the 6-membered ring, yeah, the cyclic form. Do you know how to draw the axial and the equatorial groups? How?
So the first, the topmost.
Why don't you come here?
So axial, this goes here, this goes here.
Very good.
For equatorial, it's always parallel.
No, draw, okay?
Okay.
Yeah,
So-
very good.
And for equatorial, it's always parallel to the alternate line. So this line will be parallel to this. Then again,
Yeah, be bold, go ahead. Don't worry. Very good,
this
excellent.
line will be right here.
Uh-oh. Draw every bond, yeah.
Okay, let me.
Uh-oh.
No. It has to be like this.
Well, you have drawn few bonds that are really correct and few that are tremendously wrong.
Change the bottom of
Yeah.
the
So,
Yeah. No.
this. Oh, like no.
This one is okay. This other one. The top one.
You mean this one?
No, the topmost on the left. Bring to the
Bring it out.
outside. Bring it out.
Yeah, like that.
Yeah. Just draw parallel to the
It should be parallel to this line, right? So it cannot be out.
right. Yeah,
Oh. This
no. Parallel to that. So, yeah.
Okay, maybe I'm confused.
No. You know the principle, but it's just that you have not practiced enough.
Yeah, I've forgotten actually.
Well, you're not forgotten, but you know the principle, but you have not practiced enough. So I think, first of all, you have to remember that all bonds, none of the bonds actually go in the ring. Remember that. Okay? None of the bonds go in the ring. In the ring, okay? So you can't really have, obviously this bond is not in the ring, okay? Likewise, this bond is not in the ring. So he is absolutely right. Easiest is to draw the axial. Thank you. Let's use this. So easiest is to draw the axial. Okay, so you have, these are the axial. These are the ones that are perpendicular, right? So what do? For example, if you want to draw an equatorial bond here, then this bond is going to be parallel to the, as he said, the alternate bond. So the alternate bond here is this one. So draw a line here which is parallel to that line. Yeah? Likewise, if you want to draw a bond here, alternate. So draw a line that is parallel to that bond. You're right? So when you want to, so this is correct, but I'm simply erasing. There should be, so if you want to draw an equatorial here, draw a line that is parallel to the alternate bond, which is this one. Yeah? And likewise, keep going forward. And then we have one more. This is parallel to here. This is parallel to this one. This is parallel to this one. And that takes care of all the bonds. Yeah? Are you all with me? And that looks like a nice chair. Right? Remember that. By the way, when you go for interviews and things of that sort, if people suddenly ask you, could draw the structure here, and you show up, with all this crooked, weird kind of bonds, it doesn't feel right. That means you have not practiced. And that gives away that here is a student who has not been able to really draw, not necessarily on the blackboard or the whiteboard, in general. Right? So, practice. Hopefully you don't have to draw really big carbohydrate structures. But this is how basically we draw. Now, what was I trying to answer? I was trying to answer about the axial component, right? So if you notice, these are equatorial. This is equatorial. This is equatorial, right? This is axial. This is axial. So in essence, if you notice, that and that and that, these three, they interact with each other. If the substituent is small, such as an H, then you would have not much interaction. But as soon as it becomes CH3 or even bulkier, they start interacting. So the interaction arises from the, and this is called the 1,3. This is position 1, not real position 1, but this is, for example, position 1. This is position 3. This is also position 3. So this becomes 1,3 diaxial interaction. So that's why the axial groups are sterically hindered. And the equatorial is relatively more open. And because the equatorial is relatively more open, what do you think would be the constitution in real life, in water, in solution? What would be the dominant form between alpha D and beta D? Which would be dominant?
Beta.
Yeah. In beta, you have one OH. Sorry, in alpha, you have one OH, which is axial. All of others are equatorial, right? And therefore, beta D is going to be the dominant species. Between, and I forgot to answer the question, between the open chain and the closed chain, the closed chain, the pyranose form, is going to be the dominant form. That's how nature exists. Between alpha and beta, as far as glucose is concerned, that's going to be the beta D. Okay? That's going to be the beta D. So in essence, and assuming that you have this particular OH here, okay? Let's change it. So assuming if we have that OH here, okay? So if that, not OH, that CH2OH, if this entire group occurs in this form, then what would be the dominant species? It will move over to the alpha form, okay? Because the interaction between CH2OH, that H and that H is so high, 1,3 diaxial interaction is so high that that CH2OH will not prefer to stay in the axial form. And these two forms are in, I don't have models but to show you, but these two forms are interchangeable, okay? By mere flipping of these carbons, you can actually move it around. So what happens therefore is that that CH2OH, if at all it occurs in the axial form, it will move over to be in the equatorial form. It is a dominant contributor to conformational equilibrium. These two exist in equilibrium. So a bigger substituent would not prefer to stay in the axial orientation. Are you all with me? Yeah? So galactose. So here, so galactose, for example, you would have, galactose and glucose primarily differ in the 4 position, as we have seen. So glucose, for example, has an OH here. So you would have the same alpha D galactose and beta D galactose. So if you notice that CH2OH in both cases is equatorial, the dominant contributor is equatorial, the bulkier contributor is equatorial. And therefore, despite the interaction of the OH with the H, whatever it is, it'll still stay here. It cannot be moved over. Why? Because if this moves over into the equatorial form by a flip, then that will move over into the axial form, and that will create a bigger steric problem, and nature would choose the least energy path. So this is how conformational equilibrium, in a very simple manner, is typically defined. Okay, this is how conformational equilibrium can be defined at a very simplistic level. Obviously, there are molecules for which conformational equilibrium is way more complicated, and we are going to see that in just a little while. So, just as we have glucose, we have other monosaccharides. One of them, for example, is our uronic acid. The only reason why I'm discussing is because heparan sulfate, and other glycosamiliglycans have uronic acids. And for example, in this case, the uronic acid, such as D-glucuronic acid, is formed by the oxidation of the CH2OH, which is the 6th position CH2OH. That is converted into a carboxylic acid. So glucuronic acid also has an aldehyde. Rest of the things remain the same. Nature does it in a very beautiful manner, which is why if a chemist does it, then he'll have to do a lot of different things in order to specifically convert all of that into carboxylic acid. Why? Because there are groups here that can also be oxidized. You might have to protect it and things of that sort. Now, uronic acids are important. D-glucuronic acid is an important player. Heparan has D-glucuronic acid. Likewise, there are other uronic acids that are also present. For example, you can have maluronic acid or galacturonic acid or iduronic acid. And notice the difference is iduronic acid occurs from L-idose. So this is one of the important L-series carbohydrates that is actually present in nature. Not only that, the interesting part is the iduronic acid, especially the L-iduronic acid, exists in multiple conformations. Not all of these are populated to the same extent, but exist in multiple conformations in nature. So these conformations are typically referred to as 2SO or OS2 or 1C4 or 4C1. So in this particular case, C is the chair. Okay, let's see whether I have a representation. Okay, I don't have a representation. Yeah, we could do something like this. So how are these conformations named? Okay, is what I'm coming up with. So the way that you, in order to name a conformation, such as 1S, 1C4, 2SO, things of that sort, we have a specific way in which the carbohydrate, and this is really true only for carbohydrates, okay, because of the complexity, because of, you know, the way the structures are, that we have to represent it in a specific manner. So what do? We draw the structure of the 6-membered ring such that the oxygen is always here. And the positions 1, 2, 3, 4, and 5 are in essence organized in a clockwise manner. And this is how you have to draw it in order to be able to come up with the conformation. So once you draw that and you draw an appropriate arrangement, if the carbon atom 1 is above the plane that is formed by these 4 nuclear positions, then you write it as superscript 1. If the carbon, whatever carbon is below the plane of the remaining 4 atoms, then you write it as subscript. And that's how, in that particular representation, this carbon is above the plane, this carbon is below the plane, and that's how it turns out to be 1C4. Yeah? So you can actually have 2C5 also. That's still possible. There are a lot many conformations, way beyond the 3 or 4 that I'm depicting here. Large number. In nature, a few of these are populated. Okay? And these are in equilibrium. Remember, these are in equilibrium. Why? Because you can actually flip them around. So likewise, for example, you can see that that oxygen is the same as here, for example, and these 4 form a plane. This is above the plane, this is below the plane, therefore this is 4C1. Yeah? And both of these are chair conformations. Yeah? Both of these are chair conformations. We can also have something like that. Some people call it twist boat, some people call it sofa, and there are variations in between. So this is a sofa conformation in which carbon atom number 2 is above the plane, oxygen is below the plane, and 5, 4, 3, and 1 are in the plane. And that's why this is called 2SO. And if you flip it around, you can actually get OS2. And likewise, there are several other conformations that are possible. So let's go back. And I want to show you. So we were here. Okay. So I want to show you as to how do you actually find the ground state conformation based on the open chain form. So this is connecting the open chain form with the closed form. Not only that connection, but finding the conformation of the closed form using simple principles, very simple principles, not really doing a lot of computational work. So if you look here, this is glucose. Yeah, this is glucose, okay? This is not the glucose, this is D-glucose, right? The open chain form D-glucose. So what do we do? All right, so imagine this. So what do we do? In order to convert the open chain form D-glucose into the cyclic form, so that we know which group goes where, this is what we do, okay? And you should actually draw this, but for the time being you have to imagine. So what do? We take the entire linear, so if this is the open chain form, like that, okay, aldehyde here, CH2OH here, what do? We simply put it like that, okay? You simply put it like that. So in between there is a step that I think I have forgotten, I just jumped a little bit. So in between you write it in a linear horizontal manner, okay? So the aldehyde is going to come at the bottom here, okay? So imagine, so the aldehyde is here, the OH is here, the second position OH is here, yeah? Then third OH is going to be on the top, position 3, yeah? Then fourth OH is going to be at the bottom, fifth OH is going to be at the bottom, and CH2OH is going to be here. Yeah? Are you all with me? Right? So OH, 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 and then CH2OH. Then what do? We convert it into a twisted form. I can't do it with this one. But then you move the rest of the atoms so that there is a possibility of bringing the 5-position OH, a 5 position, in a space close to CH2OH. So the OH goes at the bottom, which is already here. This OH is at the top. That OH is at the bottom. And this OH is at the bottom. Exactly the same. And then CH2OH turns up in this case. Are you all with me? Okay. Then what do? We rotate that bond so that the OH turns up in the same sort of plane as the carbonyl. So you rotate like that. So what happens? CH2OH has to go at the top. The OH will come in the same plane as the aldehyde. Yeah? And then that is basically ready for the attack on the carbonyl. You're going to get alpha or beta at this position, which we have seen earlier. So if you convert this into a form such as that, okay, what's going to happen? If you convert that into a chair form, that OH is going to fall up like that. This can be either alpha or beta, depending on where it is. I've simply drawn the beta form. Then that OH is going to be axial, this OH is going to be equatorial, that OH is going to be, that CH2OH is going to be axial. Obviously we know that this is not the form in which it would prefer to stay. Okay? So it will undergo a flip. So when it undergoes a flip, every axial orientation will convert into an equatorial orientation. Okay? I'm sorry, I just don't have models, but if you have models, you'll be able to see that. You can actually move it around fairly easily. So every axial will convert into equatorial. So this CH2OH will convert into equatorial. That OH will convert into equatorial. That OH will convert into equatorial. All of these will convert into equatorial. Yeah? Are you all with me? So, and then this is we know this is the most stable form from beta-D-glucose. But if we had another sugar that had variations in these orientations, let's say two groups were, in this case, all are axial, so it's very easy to identify. But if we had two groups, such as that OH and that OH as axial, equatorial here, then by flipping, we would have converted those into axial. So then the energetics would play a role. Which is the bigger group? The dominant feature is going to be the bigger group usually at carbon number 6. Okay? If there is none, then which is the dominant group? Which interactions are more? 1,3-diaxial interaction is more or less, and that's how conformation of these sugars are typically determined. Right? So here is how, for example, the L-iduronic acid would work. Okay? Can I go a little bit faster? Yeah. So here you can see this is D-glucose, this is L-iduronic acid. L-iduronic acid, you arrange it so that that OH comes at the bottom, that OH is at the top, that OH is at the bottom, that OH comes out at the top. Yeah? And what do we do? In order for that OH to interact with the carbonyl, you have to rotate it to the right. Instead of, in the earlier case, to the left, you rotate that bond. By rotating a bond, you don't change the stereochemistry. So you end up with something like that. You can see that the carbonyl, the carboxylic acid is at the bottom. And that's typically true for L-iduronic acid. So once we convert that, we get 1C. What happens in real life is that you actually get 1C4 form. In 1C4 form, only one OH, position 3 OH, position 4 OH, and position 2 OH are axial. Only one carboxylic acid group is equatorial. You do have 1,3-diaxial interactions here, actually several. In 4C1, you have carboxylic acid as axial, and that's the dominant feature. So 4C1 doesn't populate to a significant extent in L-iduronic acid. 1C4 populates quite a bit. But in this case, there are 3 axial substituents. So even that generates significant energy. So the nature tries to organize it to lessen that strain. That is the reason it adopts 2SO conformation. And in 2SO SOFA conformation, we don't truly have an axial or an equatorial bond. These are called pseudo-axial and pseudo-equatorial bonds. And that lessens the geometry. So the dominant conformation of L-iduronic acid turns up to be 2SO and 1C4 form. There is a small proportion of 4C1, and there is a small proportion of OS2 also present. So L-iduronic acid is actually a mixture of conformations. Very small proportions of others. The dominant ones are 1C4 and 2SO. Okay? Yeah, go ahead.
Does this change at all when it's in a polymer?
Yeah, it could change. Not only that, it actually changes quite a bit when you have different substituents. Absolutely. Polymeric interactions would also govern as to where the equilibrium lies. So, absolutely. Good question. Okay, what time is it now? 9:35. Okay, why don't we do this? Why don't we take a break? Okay, I think I'm already seeing you all are tired. All right, so why don't we take a break? I think what I'm going to do is I'm going to skip this oligos for the time being. We are going to cover oligos as we go along. I think you have learned, for most part, the absolute key fundamentals that are really needed in order to understand what's going to come up next. We have an entire lecture to do on characterization, right? And we can't really avoid characterization. So let's take a break, and hopefully we'll pick up characterization when I come next. Okay? We'll see what Kubi wants to do. Okay? Yeah? All right. So do you know there's coffee around?